Hello, my wonderful and lovely humans. It's Miss Reyes and King Triton, and we are here to teach you guys about mitosis. Yay, mitosis, yay. Okay, anyways, he can go now. So you guys should have just completed the cell cycle. Ta-da! All right, and what we are going to be talking about is this little, this little chunk right here, this mitosis chunk. So as I was saying before in our last video, there are a whole bunch of processes that occur in this chunk. We kind of cover all the other processes that occur in this one, but this chunk right here, lots of really important little bitty things are going on here. And so that's why we just need to spend a little bit more time, really focus on each one of those individual pieces. So I'm going to go ahead and flip my camera. You guys should have this assignment with you guys there. And if I can figure out how to flip my camera, give me a second. All right. Okay, so as you guys can see, I've gone ahead a little bit and I just made it really look pretty. I mean, I figure if you guys have to watch a video, at least make it look good, right? Like I said, we're going to talk about mitosis. This mitosis is this chunk right here. The rest of this is our interphase. Okay. Anyway, mitosis. Alrighty. So up here at the top, we're looking at what mitosis is and why it needs to happen in cells. So mitosis is one method of cellular division. One method of cellular division that can occur in eukaryotic cells. Eukaryotic cells. So there are prokaryotic cells and then there are eukaryotic cells. Eukaryotic cells are going to be characterized by cells that you and I have versus what maybe a bacteria might have. Okay, so a eukaryotic cell is going to be something that is slightly more complex. So it is going to be part of our cell cycle. Again, what we just finished talking about in our last video. And this is going to basically alternate, or basically it's not a part of interphase. It's a separate part of the whole cell cycle. It's its own part. But the other part of that cell cycle is that interphase here. So the creation of new cells is going to be important for multicellular organisms because it allows them to grow. It's also going to allow damaged tissues to become repaired. So in some species, for example, a strawberry plant, mitosis is going to be used for asexual reproduction. All right. And we will get into all of this nitty gritty here in a little bit. But before we do that, we need to first of all address the 4 phases that make up mitosis. And basically all those phases can be reduced down to something that we refer to as PMAT. And P is going to stand for prophase. All right, prophase. M is going to stand for metaphase. A will stand for anaphase. Uh-oh. He's here. He's here. A is going to stand for anaphase, and T is going to stand for something something we call telophase. And the king is trying to ruin my handwriting as if it wasn't already bad enough, Middleman. Okay, so you guys will see him in a second. No worries. Leave him. Just let him do his thing. All righty. So again, there are these 4 phases here, and we're going to break these steps down here at the bottom. But the first thing that we need to talk about before we can go on to each one of those 4 stages is talk about the difference between chromosomes and chromatids. So as you can see here, I've used purple to represent chromosomes and green to represent chromatids. So chromosomes, I forgot to highlight something. Oopsie daisy. And I double checked too. Anyways, I'm sure I'll find another one. So chromosomes are going to be molecules of tightly packed DNA. And oh my goodness, Triton. He's a little upset. That's his, oh no, that's his tail. So I think I gotta kick this little dude out. Goodbye. All right. Where was I? Chromosomes are molecules of tightly packed DNA that during interphase, the cell's DNA is going to replicate. So that each chromosome can be made of two sister, we stand together. I don't know the rest of the lyrics. Okay, anyways, so sister chromatids that are going to be joined together by what we call a centromere. All right, and if you guys look over here, right here in the middle, this little button looking thing, that is what we refer to as a centromere. And now I'm realizing that I probably should have highlighted that, so I'm going to do it now. Boom, there she is. All right, so they are connected together with a centromere. When the sister chromatids are separated during anaphase, all right, and if we look back here, anaphase is that second to last phase. Anaphase, we're going to refer to them once again as chromosomes. So basically what you guys need to realize is that they are only called chromatids when they are joined, together. Sorry guys, that was Alexa, and obviously I don't want to say her name because then she'll get triggered, but yeah, she's just telling me it's time for bed. But anyways, okay, so what we see down here is a little bit of a challenge, okay? So this light blue one, would we say that this is joined or is it not joined? Exactly, it is not joined. So we are going to call this a what? Exactly, this is going to be a chromosome. All right, a chromosome. Next one. All right, so if we're looking here, we do see that there are two strands here. However, if we look at this little dot, 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 we see that it's only circling one. So is this one joined together? Yes, it is, which means it must be called a what? A chromatid. Very good. All right, so here we have a chromatid. Over here we had a chromosome. So this one's a little bit tricky. Number 3, would you say that this little dude here is joined together or not? If you guys said that he is not joined together, you would be absolutely correct, because that makes this what we call a chromosome. Alrighty. So over here, all we see is this little nub or this little Cheeto puff, and this Cheeto puff by itself is called a chromosome. Over here, we see that this chromosome that is connected to another chromosome is going to be a chromatid because it is joined together. Over here, lastly, we see that this this chromosome does have a centromere, but it is not connected, right? So a little bit different there. Now, if we look over here at our diagram, we can see these little big X's a little bit closer, all right? And let's just take care of the easy one first, right? And that easy one is the center part here. And just like it says here, centromere, this is going to be our centromere. All right, and an easy way to remember that is obviously that centromere starts with like center. It's located in the center of the chromosome, okay? Now, if we're looking here, we see that we're referring to this entire body here. And when we are referring to this entire body here, we are talking about chromosomes. Okay, this is a chromosome. Now down here, because we are only talking about this one little dude, this little darker one, this is what we are going to call a chromatid. All right, And so once again, we are only talking about this little dude here, but we are talking about chromosomes together. So technically 2 chromosomes, okay? Alrighty. So now that we have this taken care of, we are going to move on to our first mitotic phase. And if we look back at our 4 phases, the first mitotic phase is going to be that P in PMAT. That stands for prophase. So we are going to talk a little bit about prophase. And let me bump my camera down here so you guys can see that all right. And here we are. So it tells us we need to doodle and describe each mitotic phase. So let's do it. So once again, this phase was known as the P phase for prophase. All right, prophase. So inside of a cell molecule, what we're going to have is a nice big cell. All right. And that nice big cell has basically what we would call kind of like a little brain. And that's referred to as the nucleus. Okay. Now, the nucleus is surrounded by this thing that we call the nuclear envelope. So think of a letter inside of an envelope. It is kind of protected in there, right? But the letter is what's really important. And guess what? It's really important inside the nucleus. It's that DNA. So inside the nucleus, the DNA is going to condense into chromosomes. And he's back. He is back. All right, so the DNA is going to condense into chromosomes. So what I want you guys to think of is a towel. If you've ever really twisted and twisted and twisted and twisted and twisted a towel, you know that eventually it really starts to shrink and bunch up. So that's pretty much what happens here in DNA. We've just got very teeny tiny little strands of DNA packed up in there. And basically what's going to happen during this process of prophase. Oh my goodness. Well, man, really? Okay. He is something extra today, guys. Extra. Okay. Seriously, guys, look at this. Look at this. Oh my goodness. Little man's got issues. Look. I don't know, man. I just wish you guys could meet him, like for real. Because like, look at this. Look. Anyways. All right. Sorry. I fashed you. Okay. Oh my goodness. Oh, ouch. Now he's digging in my neck. Okay. I think I might have flipped the camera way too fast. Oh my goodness. This is a mess, guys. Oh my goodness. Okay. Anyways, the nuclear envelope is going to begin to break down. All right. So what we got here, remember, we got that nucleus. Okay. So it's breaking down. So we're going to just kind of draw like a little dotted envelope within an envelope kind of. Okay. All righty. So inside this nuclear envelope, we have those chromosomes. And as we can see over here, a chromosome looks like this little X looking thing. Okay. So we're going to have 4 in here. And I'm going to be super simple. And I'm literally just going to draw some X's. Super simple because Miss Reyes is a biology teacher and not an art teacher. Although I would love to teach a ceramic class, but you know, we can talk about that some other time. All right. Anyways, so we have 4 chromosomes in here. And like I said, this nuclear envelope is starting to break up. So you guys can see that that little dotted line is referring to that nuclear envelope that is breaking down. So bundles of proteins that are called centrioles are going to start to move toward opposite ends of the cell. And when they do so, when they're moving to those opposite ends of the cell, those opposite ends are going to be known as the poles of the cell. So think north, south pole, right? Where you send your letter to Santa. All right, so 2 centrioles. All right, and if we look at those centrioles, what they're going to end up looking like is like some little macaroni noodles, believe it or not. Little macaroni noodles. And again, y'all don't judge me. I am not an artist. I'm a biology teacher. Yes, indeedy. All right, so here's a couple of centrioles. So when we have 2 centrioles, they're going to form together and make an organelle that we call a centrosome. All right, a centrosome. And also Miss Reyes, right? I made a boo-boo. I need to make sure that I have 2 of these because remember, these guys are moving to opposite ends of the cell. So a couple more macaroni noodles. So the good news about Miss Reyes not being an art teacher is the fact that I don't really care how bad this is, as long as you try. That is what I do care about though. So anyways, as I was saying, these 2 centrioles are going to come together and they're going to make that centrosome. So when we're referring to the centrosome, what we're talking about is both of these guys together forever. Individually, however, they are known as those centrioles, which you can see this is a slightly lighter color than this one, but they're pretty close in color. I did it on purpose. Yes. All right. So, and that is going to conclude our prophase phase. Okay. Now we can move on to the next one. And what I think we should do really quickly here is take a look at our PMAT. So we took care of P, which was prophase. Now we need to look at M for metaphase. And that is what's going to start us off back here. All right. So metaphase. Metaphase. All right, metaphase once again is our M phase. Oh, sorry, is our M within PMAT. All right, and just really quick, I'm gonna write PMAT up here just to remind myself of all of that stuff. As we go on with the rest of them, we're gonna make sure that we can refer back to the PMAT, make sure that we take care of each one of those letters, okay? So metaphase. In metaphase, the chromosomes are now going to be free in the cytoplasm. All right. So a characteristic of prophase was that we had a nucleus that started to break down. Okay. So that nucleus, like an envelope, okay, had contained the important message of DNA. And once it starts to break down, now this stuff is just kind of floating around in that little jelly that's kind of holding everything together. So what we can do here is draw our 4 chromosomes from our last picture. And again, not a science teacher, so I'm just gonna draw some X's here with some little buttons in the middle. And you guys can do the same. All right, but as you'll see here, each centrosome will have formed some spindle fibers, okay? And that's what these little strands here are called, okay? So I'm actually gonna go ahead and highlight them because y'all know Miss Reyes likes her color coding. Oh yes, indeed. So all these little orange structures over here are called those spindle fibers. And don't forget that, these spindle fibers are coming from our centrioles, or wait, centrosomes, my bad. Whoops, see, Miss Reyes is a little rusty on this. Okay, so these are gonna be coming from our centrosomes. So those were our little macaroni noodles from way back when, like AKA 2 seconds ago, right? There's a macaroni noodle and there's a little macaroni noodle, boom. And a couple more over here, boom, boom. Man, these are getting really bad. Sorry, guys. Okay, so here's my macaroni noodles, and don't forget that they were a certain color also. So I'm gonna go ahead and just make sure that I include that color. And as you guys can see, I've color coded here just to make sure that they match up. Now, I am giving you guys extra credit if you guys want to go ahead and color code everything, make sure everything looks pretty, because, you know, Miss Reyes like pretty. But you guys will get extra credit if you color this stuff in, okay? And I think this is gonna be a pretty good chunk. Just gonna say that. But if you color this in and you also color in your cell cycle, okay? And I mean like color things in, make it pretty, okay? If you guys do that, I will give you a total of 5 extra credit points, okay? And I'm pretty sure this is either gonna be a 20, 25 point assignment. So yeah, it's a decent chunk, okay? All right, anyways, so these guys, the centrosomes are going to form those spindle fibers. So those are those little strands. I like to think of them kind of as like fishing line, okay? Now that fishing line is gonna go fishing for some chromosomes, and guess what? It's gonna line up along the middle. It's going to line up along the middle of the cell, okay? And if you guys look over here, you guys don't necessarily have to draw this, but I am going to draw a little dotted line down here so you guys can see that it's kind of pretty much the center of that cell there, okay? So another way to remember this metaphase phase and to remember exactly what goes on in metaphase is to call it also the middle, okay? The middle. Because again, these chromosomes are lining along the middle of the cell. And they are going to attach to these spindle fibers through their centromeres. All right, and those centromeres were those little, and I've clearly used the wrong color here, which is my bad, y'all. Should be this color, but those centromeres are what hold the little X's, those little buttons here, okay? So that's where those spindle fibers are attaching, so those little buttons in the middle. And again, that should have been that color. Alrighty, so this is going to conclude our metaphase phase. All right. So once we get to metaphase, we took care of P, we took care of M. Now we go on to A, which stands for what? That's right. It's anaphase. So I'm going to put A for anaphase. All right. A for anaphase. So this is our anaphase phase. So here you can see we still have some spindle fibers. So let's go ahead and color those in just proactively, you know. Not gonna hurt anything. Pretty. All right, so now that we have our spindle fibers are going to contract just like muscles. They're going to contract and they're going to separate our sister chromatids. All right, so once they are separated, we're going to refer to them as chromosomes again. All right, so let's make sure that we're including that. So these guys are chromosomes here. And what we're basically going to have here is half of an X. All right, and what you guys are going to find out here in a little bit is that these guys end up looking kind of V-shaped. And again, you guys will see that here in a second. But I'll make sure that I get those drawn in here. That's the king. He's upset that I'm not paying him any attention. So here, once they're separated, we're going to refer to them as chromosomes. So these guys have now become chromosomes. They are pulled. They're going to be pulled by their centromeres. So that little center button there. So they're getting pulled by their button. And remember how I said these spindle fibers, I like to think of them as like a fishing line. Well, they're getting pulled just like you would with a fishing line, right? You catch a fish, reel it in. So pulled apart by their centromeres to the poles of the cells. And what's over here at the poles, our little macaroni noodles, our centrioles, okay? Or because they're in a pair, the more correct term would have been centrosome. But I like to call them a little bit like macaroni noodles. They look like macaroni noodles, no? Okay. So I'm also going to make sure that those colors match up with what we've been doing so far, because again, I like color coding. But yes, it just looks really pretty, doesn't it? So pretty. So much less boring than a blank piece of paper. But anyways, okay. So spindle fibers are going to contract. They're going to pull apart those X's, and they're going to make them look V-shaped here. And once again, these have gone from sister chromatids over here to chromosomes down here. Got it? Awesome. Okay. Now, real quick look back up to the top here where we wrote our PMAT. So we took care of the P, we took care of the M, we took care of the A, and finally we are on to our last phase here, which is known as telophase. So T for telophase. And there he is again. Okay, so telophase. Now in telophase, what we've got happening here is you can see that there's kind of like a little peanut going on here. All right, so what's happening here is our chromosomes are reaching the poles of the cell, and they are going to begin to uncoil. So our tightly packed DNA is going to uncoil. It's going to release itself. And instead of looking like a crazy packed in X, we're going to become longer and thinner. A nuclear envelope will begin to form around each group of chromosomes, okay? So if we look back in this picture, you can kind of see that this is starting to separate. So on this side, what we'll have is 4 little V's, okay? So make sure I got 4 little V's. I'm gonna make them point in different directions because I think that's kind of cute, but you do you. Boom, okay? And I had 4 over here and I have 4 on this side. So I'm gonna make sure that I get my little chromatids, chromosomes. See, I get tripped up. It's hard. It's not always easy. Oh my goodness, I think I did this one backwards. Oh well. Okay, so I have 4 chromosomes each. And again, a nuclear envelope is gonna begin to form, and just like we were kind of showing our nuclear envelope breaking down, well, we're going to kind of use that same ... process in order to kind of show it growing around them, okay? So it's either growing or it's forming, but because we know that this is the telophase phase, we know that it's actually growing around them, okay? All right, and here you can see that an envelope is starting to form. And what's going to happen next is that the cytoplasm, will start to divide in the process of cytokinesis. All right, cytokinesis. And cytokinesis, what's basically going to happen here is that we're going to pinch our little peanut here. And, oh, let me write cytokinesis. All right, so we're going to pinch this off here and we're going to pull them apart. Okay, and we're going to have basically 2 new cells here. Oh, and don't let me forget, but I got those little centromeres here in the middle, the little buttons. All right. Okay, so finally we move on to cytokinesis. So I realize that PMAT does not have a C in it, does it? Right? Well, that is because cytokinesis is technically a part of telophase, okay? Now, the reason we kind of refer to it separately is because it's a super teeny tiny important part. And that's basically when the cytoplasm has fully divided, cytokinesis will be complete and chromosomes are no longer visible. All right. So The reason we're able to see these chromosomes the way that we see them is because they have been so tightly packed, right? They've become dense and they have tightly wound themselves. And again, we can see them. Over here, on the other hand, now we're still going to have that little envelope over here. So instead of teeny tiny little dots, I'm going to make sure that they're a lot longer pieces because we have almost completely reformed our envelope here. And I want to make sure that that's clear when we're referring back to this. But basically what we're now going to have is teeny tiny little strands of DNA. And what that looks like when it's unpacked is like squiggles. I'm not kidding. They look like squiggles. Okay. So all this is some squiggle DNA. All right. Now, the one characteristic that you guys need to know is that these two dudes, all right, these two guys right here, this one here, and this one here are going to have the same DNA. So they are basically little clone copies, all right? They have the same DNA. So the daughter cells are going to be genetically identical. They're like twins. They're just like identical twins, but not fraternal twins, right? Because fraternal twins are not actually copies. These guys are actual copies. So they're just like clones. All right, so they're going to be identical to each other, and they're also going to be identical to the original parent cell. All right, now they are going to be in the process. So as we are down here, oh, and I forgot to write cytokinesis. Got a little ahead of myself. Anyways, as I was saying, so while we are in cytokinesis here, they are going to return back into the interphase process. All right. And we will begin to prepare for the next round of mitosis. So remember, this is a whole cycle, right? And cycles, all they do is they continue to go around and around and around and around and around. Okay. Now, real quick before we end this, I just want to point one thing out. So if we look here at our interphase, right, once we've completed mitosis, we're going to go right back into it. All right. So depending on whether or not we have favorable conditions, depending on whether or not we have enough food or water or shelter or whatever it is we need, as long as we're not trying to just survive, what we're going to end up doing is continue this process. But if we do need to take a break here, we will go into our GO phase. But as I said, over here, we have that interphase process, and it's going to take up pretty much most of the pizza pie here. Just a little pizza slice over here is going to be that mitosis. But as you guys found out, mitosis is got a lot of stuff going on here, right? There is quite a bit of stuff going on here in mitosis. And it's not just the back page, it's like the whole front page too. All right. So I am going to give you guys a couple quick shots. And of course, you guys gotta see the king here. Say hi, say hi. No, okay, well, he's a little bit camera shy right now, which, you know, who could have thought? But anyways, I'm gonna give you guys a quick flash of the full page. Well, not really. Seems like it's too big of a paper to actually completely fit on there. But hopefully you guys can see that a little bit. If you guys need to take a screenshot really quick so you guys make sure that you fill everything in. Once again, I am offering extra credit if you guys decide to fill things in, make it pretty, make that nice and colorful. Do as much as you can, because why not, you know? And here's that quick view of the back here. So hopefully you guys can also get that filled out. Okay. All right. So that kind of concludes our whole video, and King Triton has yeeted himself out of here. And I think that means it's kind of time for bed, which he's probably not wrong. So I hope you guys have a wonderful day. Please, please, please be safe, have fun, but be safe. Okay. I will see you guys tomorrow. I love you. Tomorrow.